this International Virtual Learning Series, 43rd International Virtual Learning Series. Today we are very much delighted to welcome our eminent personality, a well-known academician and honorable resource person, Professor Dr. C. Kumarappan, School of Pharmacy, Department of Pharmacology, School of Pharmacy, King Khalid University, Saudi Arabia, to grace this occasion for his kind consent to share his knowledge, experience, and expertise to deliver the lecture on clinical pharmacological importance of andrographics and its constituents in the management of viral infection. The expectation of a traditional treatment. In the 43rd International Virtual Learning Series, in association with the Indian Pharmaceutical Association, South Tamil Nadu local branch, head office at Almi College of Pharmacy. It is a great pleasure. It's great pleasure for all of us that he could find time to be with us in spite of his busy schedule. I take this opportunity to convey my sincere thanks to you and your esteemed university for accepting our request despite of your busy schedule and for being the valuable part of this series. Most welcome, Dr. Kumarappan sir, Professor Kumarappan sir. Most welcome you, sir.
Thank you very much for your introduction, sir.
Yeah. Dear students, kindly utilize this opportunity to explore your knowledge. This is the first and foremost needed for your career. So kindly utilize this great opportunity. All the very best for all the students. Once again, I welcome you all. Thank you. Thank you, Professor. Thank you. Please,
Thank you,
you
sir.
continue. Please continue.
So, before I start my session, so I would like to I must thank Dr. Venkateshan, sir, Principal AKCP, and Dr. Vijay Panti, Dr. Rajapati for approaching me to have this series of seminars for the benefit of students. I hope at the end of the seminar, the students will get some useful information which is related to your academic interest. So with this small introduction, I would like to begin my topics. So before I start my topics, I would like to talk about what is the content of these topics. What I'm going to talk about next maybe 1 hour. So basically that's a small introduction I wish to give to everyone. So before I start my topics, why I choose these particular topics, because we have been currently encountering the viral infection currently. We are talking about a lot of synthetic drugs and many opportunities for the development of drug discovery. But sometimes we forget about our own natural resources like biodiversity. We have a lot of herbal medicines. Is very
Sorry to interrupt.
Yes, sir.
Voice is not clear.
It's not clear, sir.
Okay, sir.
It's clear now.
Yeah, you can proceed, sir. You can proceed.
Yeah. Can I proceed?
Audible.
Oh, okay, sir. So I think it is time to talk about our own natural resources, what we have, and we can use this kind of medicinal herbs for the development of natural leads for the new drug discovery and for the formulation and development. That is the reason why I took this particular plan to talk about for this seminar. So in this topic, I'm going to talk about two major aspects. I'll be talking about what are the pharmacological studies have been done so far on these particular herbs, and what are the clinically, how the clinical trials has been done to develop a formulation for mainly to address the viral infection and to boost the immunity. This is the basic idea of my talk. I'll be moving to the next slide. So the lecture outline. First, I'm going to talk about ethnopharmacology. What is the herbs and what is the constitutions? What is the traditional uses, and about the phytochemistry. This is the first talk I'll be covering the topic. Then I'll be moving to the what are the antiviral mechanism, what is the immunopharmacology mechanism has been proposed based on the pharmacology study has been done so far. This is the second aspect which I'm going to cover in this lecture. Then I'm going to share some of the clinical trials has been done so far from a different part of the countries, different part of the world, start from India or many countries. They've done so many clinical trials to bring these herbs into the market, into the commercial preparations, and finally I'm going to talk about challenges. Though we have a lot of trials has been done, a lot of patents has been registered in the name of Andrographis, and there are so many commercial preparations available in the form of supplements, but still we don't have any pharmaceutical compound. We don't have the pharmaceutical compound from the Andrographis paniculata. Why still we have problem in this? That's the last talk I'm going to cover. Let's start with the ethnopharmacology. Okay, I think you know what is ethnopharmacology. You know about pharmacology. Ethno means the traditional uses. Okay, we talk about in this first part of my talk, I'll be talking about ethnobotany and how the plant is distributed, and what are the phytochemicals has been isolated so far from this natural plant, and what is the medicinal uses has been reported. Start from the Ayurveda and from the different traditional medicines around the world. So this is herbs. Basically, it's a small herbaceous plant. It's not a big tree. It's a small herbs, and this is the herbarium has been stored in the after preserving the herbarium, how it looks. Those who are really interested in pharmacology, you can go back and read a lot of information about ethnobotany. This is some basic facts, fundamental information about the Andrographis paniculata. So the species name we call it Andrographis paniculata. So it's kind of niche family. I mean, we call it Nilavempu in Tamil. It's called Nilavempu. So synonyms: Justicia paniculata is a synonym. Sometimes you can also find some literature based on this name, but most of the literature you will find in the name of Andrographis. Okay, this is the most common name used to find the literature. It is commonly known as a king of bitters. It is the highest bitter. It is because of the triterpenoids. So this particular compound has a lot of terpenoids, basically diterpenoids. So that's the one which contribute for the bitter taste of this plant. Okay, so that's why we call most commonly king of bitters. The family, if you talk about the family, the Acanthaceae, this is a family. It's a type of plant. It's a herbaceous, very small herbs. It grows very everywhere, and the distribution is mainly in the southern and southeastern Asia. So you will find all over the Asia countries, start from India and Java and Sri Lanka, Pakistan, Indonesia, all the southern Asia. Now there are many countries started to cultivate these herbs. Like for example, Nigeria, they started to cultivate these herbs, and even some of the tropical America places they started to cultivate. Hong Kong, these are the new countries they started to cultivate, but it is mainly distributed in India and southern Asia. This is some of the basic facts. I mean, fundamental information about the Andrographis paniculata. But if you wish to know more about it, you can refer the Indian Pharmacopoeia. You will find a lot of information. So this is the distribution for these herbs in India. So you can look at the picture. Almost it distributes about 80% of the country is well distributed. So this is the close picture of the Andrographis paniculata. This is the flower. Just move on to the next slide. So I wish to talk about some of the medicinal uses has been indicated from the different medicinal system of the different country. Let's start from the Indian traditional medicine. So this has been mentioned in Ayurveda, Unani, Siddha, and homeopathy and other folk medicines. Indicated for many pharmacological applications. I will show you later. And it is also most commonly used in traditional Chinese medicine. So China is the one of the top country is using these herbs very effectively when compared to the India. It is also mentioned in Japanese traditional medicine. We call it a kampo. And it is also mentioned this about the herb in traditional Malaysian, traditional Malay medicines. It's also mentioned in Thai traditional medicines. They're using these herbs for the different purpose. Then we also Scandinavian traditional medicine is a Western traditional medicine. They're also using these herbs. That's why some of the countries like Sweden, they have done some trials on these herbs. They brought some product in the market. So these are some of the traditional medicines about the information you will find about these herbs. But still, we don't have very much information about the modern medicines. Okay, still a lot of research going on to bring into the market. So this is the different names as we mentioned, like a Sanskrit, it's called Kalmega. It's the most common name, Sanskrit name. And Spanish is called Andrographis. In Tamil, it is called Nilavembu. You can see here, it's a Nilavembu. So these are the different types of traditional medicine has been used all over the world. So this is the indications, okay. This is the ethnomedical indications. It's not the reported one. So ethnomedical information. So in Ayurveda, it is mentioned to use for fever and also liver disease. That is why we have a lot of literature on the hepatoprotective about the Andrographis. And in Japanese traditional medicine Kampo, they mentioned that the common cold. In Malaysia, they are using for mainly for diabetes and hypertension. And Scandinavian traditional medicine, they are recommended for fever and common cold. And traditional Bangladeshi medicines, they use it for many purposes. I wish to highlight some of things where is related to our topic, common cold, and also indicated for pharyngotonsillitis and sinusitis. So these all related to the infection related. That's why I highlighted this one. But look at all the traditional medicine, they are mainly indicated for the influenza virus, mainly in the common cold. That's why these herbs has been commercially, they are using as a supplement in the form of immune stimulant. And next, I wish to share about the phytoconstituents, the phytochemicals of the Andrographis paniculata. There are two major phytochemicals has been identified from these herbs. So the first class is called diterpenoids. Diterpene lactones, we call it diterpene lactones. It's basically a terpenoid class of compounds. Then we have the flavonoid class. So we call it a phenolic compounds. They have the hydroxy groups. So as you can see, the first one. So for example of the terpenoid class, I can give you the major constituents. Okay, we call it the Andrographolide. This is the diterpene lactone has been isolated first, and most of the activity is based on this phytochemical. Most of the activity is reported based on this phytochemical. There are some flavonoid compounds has been isolated from this plant. We call it Andrographidin A. We have G. So we have many compounds. So they have the phenolic compound. You can see it's a phenolic groups. Okay. So this is the phenolic groups. That's called flavonoid. And this is the typical structure of Andrographolide. So all the naming has been given, number has been given. Basically, it's isoprenyl unit. So I think you might have studied in the chemistry. So what is known about the isoprenyl units? So they have the double bond. So this is a very important functional groups required for antiviral activity. So this is the major active principle. So how much percentage? About 2.39% has been helps contain this much of Andrographolide. This is the percentage of the constituents. So apart from the Andrographolide, there are more than 50 Titerpene has been isolated. Apart from the 14-deoxy-andrographicide, we also have neo-andrographolide, andrographicide. So there are many andrographolide. You will find all the literature has been published in this article. So triterpene basically is a secondary metabolite, same like alkaloid, same like a flavonoid. They contain 20 atoms of carbon derived from the condensation of 4 isoprenyl units. That's what I said before. Triterpene mainly contain the isoprenyl units. So let me explain what is isoprenyl units. I'm not going into very deep in the chemistry. Just want to give you some basic information. So basically, they call 2-methyl 1,3-butadiene, branched chain, unsaturated chain. You can see here, it's an unsaturated chain, hydrocarbon chain. Basically, they are unsaturated, and they contain one or more double bonds. You can see here, they contain the double bonds. That's between the carbon atom. This is called the definition for the isoprene compound. Okay, so this is the one of the 4 isoprene compounds. They condense together and they form the andrographolide. This is the compound they contributing majority the pharmacological action for these herbs. All right, that's all about some basic ethnopharmacological information and phytochemical. Let's move on to the how Andrographis has been tested for antiviral and immunopharmacological activity in terms of pharmacological experiments. We are going to talk about the Andrographis and with the viral infection and for the general immune functions. Before I talk about the antiviral and immunomodulation activity, so just to know some of the other activity has been done from these Andrographis. This particular herb has the overwhelming study has been done. I mean, extensive literature has been done, and so many pharmacology has been done because of the nature of the herbs. Most commonly, it has been tested for hepatoproductive and is still used for the anti-diabetics traditionally. That it has been proven anti-diabetic protein, hypolipidemic, so it can be used for some of the malarial parasite, anti-malarial, and it is very effective for the gastroproductive anti-ulcer agents and cardiovascular function and neuroproductive function, antidepressants. So there are so many pharmacologists done. This is only a very small list of study I just shown here. But those who are interested, please, if you are specifically interested, you can find more information on the literature. So all the activity has been based on the andrographolide. So that's a key cheap active principle which has been reported in the Andrographis. Let's move on to the antiviral effect. There are a number of viruses has been tested for these Andrographis. Start from the based on the traditional literature. So most commonly reported for a common cold, influenza A virus. So there are a lot of literature has been done, and pharmacology it has been proven the herbs has been very effective for influenza A virus based on the ethnomedical information. Now a lot of research is going on, and some of the research has been proven the mechanism how that is effective against hepatitis B virus, HPV, and some literature says that it has been tested for HIV-1, okay, even immunodeficiency virus. And I think you know about we use these drugs for the dengue virus as a decoction. People, even the government of Tamil Nadu recommended to consume the herbs for the dengue virus, especially serotype 1, T and V1, and it is all used for the HPV, papillomavirus 16 strain, and used for the Herpes simplex virus, HSV, and very recently we used the chikungunya virus, CHIKV virus. It was very effective for the killing the virus virus particle. Now many researchers interested on COVID. There are some literature has been published, but still it's not in the experimental level. Still on the in silico level, they are working on this active principle. They are docking with the virus particle. Maybe we can expect more research or in upcoming years about particular COVID virus. So this slide will explain about what is the general antiviral effect of andrographolide. Okay, particularly I'm talking about andrographolide. There are 4 major mechanisms has been general mechanism has been proposed. I just summarize here. The drugs blocks the viral entry of the virus. That's the first stage of virus penetration. So the drugs proven blocks the entry of virus, then it blocks the viral protein synthesis. It's one of the very important process of the viral replication process. Then it is also blocks the replications of viral replication, and finally it blocks the protease enzyme. The enzyme cleaves the viral protein and release the virion particle. Okay, these are the four common mechanism of the Andrographis, and its constitution has been reported. And I can go individually. As you can see on the picture in the right hand side, I start from here. It can be tested for chikungunya virus, chikungunya virus. It blocks the protein synthesis. It inhibits the HSV1 and HV, and it also blocks some of the viral receptor signaling process, and it promotes the interferon. Can you see SCV? That is C virus. It promotes interferon gamma, because interferon gamma is one of the powerful immune modulator. It can kill any virus. So it's particularly effective for the HCV virus because the Andrographolide, which stimulates the production of interferon gamma. And in the HIV, it increases number of CD4 cells. The lymphocytes, especially CD4 lymphocytes, the number has been increased after consumption of Andrographolide, especially those who are infected, HIV infected patients. Then you talk about the HPV, papillomavirus. So it blocks the one of the modulator E6 oncogenic protein expression will be blocked, and some we also know that some virus can cause the cancer. Okay, HPV, because cervical cancer, human papilloma virus can cause the cervical cancer. So in that, so Andrographolide increase the P53. It's one of the tumor suppressive genes. So it restores the P53 gene so that P53 can block the progression of cancer in cervical cancer. This has been reported. And also reduce the RNA copy of the number of RNA copy of the chikungunya virus, so that the replication will be reduced, the viral count will become reduced. This is how the Andrographis is very effective in the chikungunya virus, because it reduces the number of RNA copy, and it is proven against the HPV, HSV, and they stop the DNA replication process. I mean, they blocks some of the enzymes involved in the DNA replications. Then it blocks the HCV, hepatitis C virus. It blocks the protease enzyme. So this is the fourth mechanism. It blocks the protease enzyme. Then Epstein-Barr virus, because the protein express will be stopped. Finally, herpes simplex HSV is called herpes simplex virus 1. It blocks the glycoprotein, because these are the modulator. Okay, modulator normally required for expression. So those protein will be blocked. Expression of these protein, glycoprotein will be, viral protein will be blocked by the Andrographolide. It is also blocks some of the entry of virus, especially herpes simplex virus. Entry will be blocked inside the normal human cell. So these are the different mechanism. So all the mechanism basically fall into these four category. Either they blocks the entry of the virus, blocks the protein synthesis, and blocks the replication, and blocks the protease enzyme. Except there are some which is called increase the interferon gamma response. So it is very useful for the immunomodulations. Yeah, this is the just quick summary about the antiviral effect of Andrographolide. Those who want to get more information, please refer the literature. And this is how the cycle or virus replication cycle. It blocks the first one, virus entry here. So Andrographis is known to block the viral entry in herpes simplex virus. Then it blocks the replication process. Okay, yeah, different virus has been proven. Okay, it has been tested on the different virus. It blocks the replication process of the different virus, like chikungunya, hepatitis C virus, hepatitis B virus, and herpes simplex virus. Then it also blocks the number 3 protein synthesis. Blocks the protein synthesis of the herpes simplex viral protein synthesis. And finally, it blocks the protease enzyme, the enzyme which is required to release the mature protein before the infections. So that's called protease activity will be blocked in the different virus. This is a very general mechanism. Next, I'm going to talk about the how Andrographolide it play a very important role in modulation of the immune function. I mean, how it stimulates the immune system. Because based on this mechanism only, there are a lot of herbal supplements has been available in the market. Because these are the mechanism how Andrographolide can be used to stimulate your immunity when you have the infection or maybe for general health. If you want to improve your general health, so you can consume Andrographolide just to stimulate the immunity. So the first mechanism, so after taking the Andrographolide, it increases the proliferation of lymphocytes. So I mean, especially T lymphocytes, the number of T lymphocytes will be increased. So it is very essential for fighting against the infections. And it also increase the interleukin-2 production because this is also part of the immune system. Okay, when you have the infections, you need to have more cytokines to fight against the virus. It also promotes the lysis of the cancer cell. In some of the cancers they found, it promotes the killing of the cancer cell by causing the lysis through NK cell. So NK cell is another part of the immune cells. NK cell is another part of the immune system. So immune cell which can kill the cancer cell through the lysis process, and also it has an anti-inflammatory because it reduces all the pro-inflammatory mediators like cytokines. We call it the cytokines. It's the proteins which cause the inflammations, mainly cause the inflammations when you have the infection or when you have the autoimmunity or anything. Example, interleukin-1 beta, interleukin-6, and granulocyte colony stimulating factor, okay, and TNF alpha. These are the very important inflammatory. So all the pro-inflammatory cytokines level will be reduced when you have the infection or maybe other inflammatory conditions. That's why we use Andrographolide for the rheumatoid arthritis and for the hepatitis infections. It also activates another transcription factor, which is essential for the proliferation of T cells. So Andrographolide known to activate the nuclear factor, transcription factor, so that a more number of T cell and more amount of interleukin-2 will be produced when you have the infections. And it also stimulates the production of cytotoxic T lymphocytes. So these T lymphocytes can directly kill the virus particle. So these are the very general mechanism, or has been proposed based on from the pharmacology studies, and based on this mechanism. So what could be the pharmacological indications we can use for anti-cancer? Because the inflammatory process is one of the mechanism has been proposed for the cancer, and it can also use for the immunomodulation, immune stimulant. I mean, if you want to stimulate your immune system, so you can use the Andrographolide based on this mechanism. And if you have some kind of autoimmune disease, for example, arthritis, it's a very powerful anti-inflammatory agents because the drugs which reduce all the pro-inflammatory mediators, which is play a major role in the inflammation. So the application is very wide. So this all the mechanism based on these indications. Let's go into the mechanism. I just don't be afraid by looking at the picture. So this is the basic mechanism of the immune system, how it works. This is the T cell. So when there is infection, all right, so your Andrographolide will activate the nuclear factor, as I said before, okay, NF-AT. This is the nuclear factor. Normally it will be inactive form when you don't have the infections. When you have the infections, or maybe when you have the inflammation, or in other conditions, this NF-AT will be activated. Normally you will be having in the cytosol. This is a cytosol, this is the nucleus, this is a T cell cytosol. So normally it will be in the cytoplasm. So then it become activated by specific a protein. Then this will come inside. This will cause the transcription. Okay, that means this the gene will make IL-2. IL-2 means the interleukin-2 will produce too much. Then this will go and fight against the infection. Similarly, the number of T cells will be increased. Proliferation will be increased with the help of this Andrographolide. This is the proposed mechanism how Andrographolide which stimulate the NF-AT, and how NF-AT which increase the production of IL-2 by activating the IL-2 genes. So this is a simple mechanism. So many of the immunomodulated drug will work this same mechanism. If any drug want to increase the IL-2 production, or any drug want to produce the T cell number, increase the number, they have to work on this similar way. So what I'm trying to say, so Andrographolide also will following the same mechanism. So they will finally activate the T cell receptor. So more interleukin will be produced, more number of T cell will be proliferated, which is good enough for the fighting against the infections. This is a quick summary about the how Andrographolide is actually produced a mechanism. I just summarize here. The first mechanism is increase the interleukin 2 is one of the very powerful cytokines which work against the infection. Then it increases the number of cytotoxic T cell, T lymphocyte, CTL, cytotoxic T lymphocyte, and they increase the nuclear factor activating factor. So it is essential to produce a more amount of interleukin 2. Then it increases the JNK, which is also part of the NF-AT activity, which is cause phosphorylation of this and JNK activity. It also contribute the anti-inflammation. So finally, they also reduce the pro-inflammatory cytokines like TNF alpha, very important cytokines. All the pro-inflammatory cytokines level will be reduced too. So this is the proposed mechanism, immunopharmacological mechanism, or I can say is the immunomodulatory mechanism of Andrographis paniculata and its constituents. Very recently, after the pandemic, we have the COVID. So one of the institute from the Chennai, I think from the University of Madras, they tried to do some kind of docking with the SARS protease enzyme. How Andrographolide, is there any correlation with the Andrographolide? Can we try to develop as a drug for the SARS COVID? So one of the attempts has been made and it has been published in a couple of months before. And the result says that they took the Andrographolide. So this is a 3D form. They took the protease enzyme of the SARS-CoV enzymes, is a target. So this is the ligand. So they try to dock, and this is how the binding pocket of the protein. So what is the conclusion from this research? It has been published just 2 months before from the University of Madras. So the molecule obeys Lipinski rules. I think people who are working on this area, they know what is Lipinski rules. And as a pharmacy student, you'll be studying the chemistry about the Lipinski rules, which make it a promising compound. They're saying that the result says that it is a promising result. I mean, it exactly bind to the COVID protein. It could be, we can develop as a drug molecule, and but we need to test before the biochemical and cell-based assays to explore its potential use against the COVID. So if you want to develop as a drug, you must test in the cellular system. Maybe you can isolate the virus. You can test the compound in the laboratory. Then we can explore against this COVID infection. This is the recent literature has been published. You will find more detail in this website. So that's all about the. So what are the? I just discussed about the mechanism, how Andrographis and its constituents has been working as a immunomodulatory agents or maybe antiviral agents for the different infections. So I'll be moving to the next topic. I'll be start talking about the next topic. So what are the clinical trials has been done so far against the infection? Because since our focus is more on the infectious disease, so especially we are going to talk about the respiratory tract infection. I'm not going to talk about the other infections. So what are the trials has been based on the epidemiological information, based on the pharmacology study has been published. So how many trials has been done, has been attempted made to develop a pharmaceutical compound. So I'm just going to share some of the information. So the number of countries performed the clinical trials. So these are the countries are given here. So start from the China has been done many number of study. Okay, they have done extensive clinical trials. That's why there are a lot of pharmaceutical compounds is available in China, so not in India. So there are many compounds in the form of injection, in the form of capsule, in the form of tablet available in the China, because they have done quite extensive clinical trials. They have proven the Andrographis and its constitution is very powerful immunomodulatory. So more than 12 trials has been clinical trials been done, really remarkable, and followed by Sweden, and they've done 2 clinical trials, and Russia has been done 2 trials. But look at India has been done only 1 trial so far has been done, and having such a biodiversity, and it is actually part of our medical information in Ayurveda. It is we mentioned so much about the Andrographis, and so this. But if you look at the clinical trials, it's very limited. Just only 1 trial has been done so far. I think one of them from the company from the Bangalore, they made attempt, they succeeded, and they brought the product. It's available in the market right now. But when compared to China, we are too far away from the China in terms of clinical trials. So we need to have more trials to be done to develop the Andrographis, the pharmaceutical compound, and followed by the Chile. There's a 1 trial. Thailand, they have done 1 trial. There are many countries has been done. This is just a small piece of information which I can share with you. So if you have more detail, you will find about the information about the trials detail. I mean, how many patients they used, what is the inclusion criteria, what is the exclusion criteria, I mean, what is the outcome of the study, so what is the limitation of the whole things you will find in this article. So elaborate information you can find. And all the trials, so what are the trials I mentioned there, they focused the clinical indication. These are the clinical indication they tried to test on the human patients. They're mainly looking for upper respiratory tract infections, including the acute one. They didn't go for chronic one. They mainly tried for acute upper respiratory tract infections. Start from the influenza. They tried tonsillitis, acute tonsillitis. They also tried for the pneumonia, acute bronchitis, pharyngeal tonsillitis, herpes simplex virus. Okay, anginosus is a kind of streptococcus species which will activate the herpes virus, so it will reactivate. That's why it is called herpes anginosus. Streptococcus anginosus, it will activate the herpes simplex virus, bronchiectasis, acute bronchitis. Then Cox virus. Okay, they use upper respiratory infection, some virus call. Okay, so maybe in future we can expect some company or they may start the clinical trials for the by using these Andrographis for the coronavirus. Yeah, okay. So there are many trials has been done. So all the trials has been very positive, and the outcome was really good. So they indicated for. So after the trials, they indicated the product for anti-infections, anti-cough, antipyretics, antiviral, and immunomodulatory. These are the indication after the clinical trials has been done. So I wish to share some of the information about the company and what is the product and what kind of form and which country they involved. Okay, these are the formulations has been done clinical trial and from India and other countries. I just start with from India called Comcore. So it has been done by Natural Remedies from the Bangalore. So they developed a capsule. So you will find the trial information from here, and they tried for the uncomplicated upper respiratory tract infection. So this is the brand name, Comcore. So from the Natural Remedy, you can get this herbal formulation from commercially. We also have the lab app from the Thailand that developed a capsule. They tried for pharyngeal tonsillitis. Then we have the Kanjang from the Swedish Herbal Institute. They tried for tablet for the common cold. So the company, I mean, these are the formulations. So these are the different pharmaceutical company. They brought the Andrographis into the tablet form and liquid form. Some they tried the granules. Okay, so all the Chinese name. So the Chinese are doing really good job in terms of in Andrographis paniculata. So those are some of the information I just want to share with you. So how important Andrographis So that's why we have to focus more on the drug development process. So, okay, if you're really interested in academicians or maybe industrial, you can take these herbs or maybe constituents. You can start developing the lead molecule. So for this is my final topic. So I'm going to talk about what are the challenges on drug discovery and drug delivery system we are facing currently about the Andrographolide. Okay, I'm going to talk about the limitations. I'm going to talk about some of the attempt has been made to make a semi-synthetic compound. Then I'm going to talk about some structural activity relationships. So if you change the structure of the compound, so what would happen? Okay, so that the activity may increase or decrease. Then what are the formulations? So what are the new strategy we can bring to increase the possibility of drug development from the Andrographis? That's my final topic. Let's start from the limitations of Andrographolide based on the new drug delivery or new drug development. Why we could not be successful to develop the lead molecule from the Andrographolide? So what are the limiting factors? Why the pharmaceutical companies not interested in developing the drug development from this natural compound? Okay, there are certain rate limiting factors. Okay, so clinical studies shown, some of the studies shown, they come to know that the drug has very poor oral bioavailability. Okay, I mean only 2.6%. It is really very poor. So you cannot go to the next stage to develop a drug based upon this data. So that is one of the areas that we must address to improve the bioavailability. That is a very important factor as a drug. If you want to develop a drug, as a pharmacokinetic parameter, so you need to focus. The second important pharmacokinetic parameter, solubility. So the drug has a very aqueous solubility. Let's look at how much, 3.29 microgram per ml. This is really very poor. That's why the drug still we have problem, because the solubility is another major issues. The third parameter I wish to talk about, the drug has extensive first pass metabolism. The drugs undergo extensive first pass metabolism in both intestine and the liver. So any drug which has the first pass metabolism, I think you might have studied pharmacology, so it's going to be trouble. It's going to be trouble in delivering the drug, because most of the drug will be inactivated before reaching the systemic circulations. So this is another rate limit lead molecule development. Another fourth important parameter, so that has the potential drug possibility of potential drug interaction or maybe potential herbal drug interactions. Because why? It induces one of the cytochromes, specifically the type A form, but it's not the primary. But still, we have to be concerned about this action, because this induction can lead to potential serious drug interaction. I will share what could be some example I will give you later. The last one, so the drug has the poor penetration efflux, because we have the peak glycoprotein. This protein can easily expel the drug from the entering of the drug into the cells. So permeation is very poor permeation. So this all the character with this character, we are unable to succeed unless if you address all these parameter to improve this parameter, then really we can succeed in making a pharmaceutical lead molecule. Okay, so these leading to very poor therapeutic application. That's why many of us not interested to work on this Andrographolide. Yes, I'm going to talk about how Andrographolide. So what happens if you slightly change the structure of the molecule in terms of if you want to make a semi-synthetic compounds. If you try to attempt a semi-synthetic compound, you must remember these are the basic rules before you try to make, especially for the antiviral. If you are going to work on the antiviral in the in silico, please remember these all the things. Okay, you must before making the changes. So this is the general structure of Andrographolide. So the numbering has been given from here. So let's start from the first one. So what are the things essential to increase the antiviral activity? This is, they took example for the hepatitis they took the compound Andrographolide, they tried to change the structure. So, for example, these hydroxy groups crucial for anti-HPV, anti-hepatitis B virus, and also this. These two groups are very important for, very crucial for if you want to fight against the hepatitis B virus. If you try to change these two groups, eventually you will lose the activity. The activity will be reduced. Let's say, for example, if you try to do the oxidation, the compound will, these two functional groups will be lost. You will lose the activity, so it will be demolished. Or if you do the epoxidation in the 17th position, also which leads to reduce the antiviral activity. It is about the antiviral. And next one is double bond. I said isoprenyl unit is very crucial for antiviral activity. Okay, if you try to remove this isoprenyl unit, especially the double bond here, if you try to remove this one, then it's going to be a problem. Okay, this is very essential for the to retain the anti-HPV activity, to improve the activity. So to try to change this functional group, yeah. But this position has no influence on the anti-HPV. Okay, I mean you can try to change it, or can try to remove or anything do. It's not going to affect your anti-HPV activity. It's not going to do anything. The next one, if you try to work on the anti-cancer compound, if you develop some semi-synthetic compound from this Andrographolide, so these are the functional groups, especially functional groups 14. Very essential. It is very essential for the chemo selective activity, so anti-cancer activity. These hydroxy groups is very essential for anti-cancer, and also this alkylidine compound, okay, beta-lactam group is very essential for anti-cancer. So it is up to you what kind of activity, what kind of pharmacological activity you are looking for. So you have to make it changes. So if you are working on anti-cancer or maybe antiviral, so these are some of the fundamental things you have to remember when you're going for structural alterations, structural active relationships. I hope you got some. I'm not really good on the chemistry, but I just try to explain some basics about the how a structural activity relationship will improve or will reduce the activity of a particular compound. So finally, I came to the what a strategy we can make based on the things we discussed before. Like I said before, the drug has very poor oral bioavailability. It has very poor lipid solubility. It undergoes efflux. Okay, it is expelled by the P glycoprotein. How to address? So there are 6 strategy has been proposed based on the literature. It's just summarized here. The picture can illustrate. The first strategy, we want to improve the solubility. Solubility is a very, very important parameter when you want to develop the drug molecule. And because this particular drug has a poor solubility, so you must improve the solubility by making some kind of liposomes, or maybe like other kind of maybe other formulation. You have to change the formulation so that you can improve the solubility. Then you can try to improve the modified drug release. Okay, the drug can undergo extensive first metabolism. Maybe you can go for the nanoparticle. So this is strategy we are moving. The first strategy to improve the solubility. The second strategy to modify the drug release. You want to release the controlled drug release. Maybe different. You can go for the different nanoparticle preparations, or you can use the micelles. Okay, like emulsion kind of things. Then finally you can improve the bioavailability. So making these all the changes, so eventually you can improve the biosolubility, which is you can see the down. So then you can also improve the permeation. There is another important factor which limits the drug development of the Andrographolide. So efflux. So you have to address the membrane permeation. So you have to improve the drug entry of the drug into the cell. Okay, virus cell or maybe anything. Okay, that is very important. So you can achieve by neosomes, or you can go for the silver nanoparticle. Okay, silver nanoparticle SLN, or you can go for the metallic nanoparticle. You can attach some metallic nanoparticle, so you can go for active targeting. Then once you address all this problem, then you can go for the target. You can specifically target what virus, what is the actual protein, what gene you want to target. All will contribute not only also increase the bioavailability, it is also helpful to reduce the dose of the drug. when you go for nanoparticle, you can target delivery, so you can minimize the dose of the particular drug. So this all the current attempt has been going on, and we are expecting the new approaches like upcoming nanocochelates is a new approach. So you can try cubosomes. So we are working on the liposome, nanosomes, but now it's cubosomes. Ethosomes are the new formulations coming up. So you can try to make some attempts to improve the solubility, to improve the drug release, to improve the bioavailability, and eventually you can come up with some kind very useful pharmaceutical compound. So you can use this strategy. The next thing is safety. We always talk about the positive, but sometimes we never talk about the safety. Why safety is very important, especially when you're going for Andrographis for long term. So you must consider safety of the herbs, especially when you go for formulations. So as far as based on the literature, it has low toxicity when used appropriately. Unless if you're using high dose, then it may cause toxicity. There is one serious toxicity has been reported based on the clinical trials. It has been proven already. A drug which has a serious skin reaction and anaphylaxis. This is one of the things very important you need to remember. But this will happen at the high dose only. So you need to worry about if you use a hollow dose, nothing to worry. This is because of if you use the methanolic extract, for formulation and development, then there is a possibility of hypersensitivity reaction. So this you need to remember. This is proven in the humans. But in terms of preclinical toxicity, got some information from the European Medical Agency, so that we know the LD50 40 grams is too high, and no genotoxicity data has been found. So unfortunately, it doesn't cause any. It does not going to affect your genes or any genotoxicity. But there is one toxicity has been reported, reproductive toxicity. Okay, that's why we use Andrographis paniculata for anti-fertility effect. So a lot of literature says it's a very potential compound to have the anti-fertility effect, because it reduces sperm count and motility and disturb the spermatogenesis process, but it is only done in the preclinical stages. And also they found that CYP2 induction, possibility of drug interaction, especially with the theophylline, because theophylline has been metabolized by the CYP1A. If the drug induces this one, the theophylline will be metabolized quickly, and you will not get any therapeutic efficiency. So these are the preclinical toxicity you have to be remembered when you're trying to develop your drug formulations. So there are some country has been developed, a regulatory document to have the safety about the Andrographis and about the anaphylactic reaction. So you can refer some document. Okay. So I think so far Australian government has made some safety regulations, so it's available in the website. But I think we don't have from India, we don't have any safety regulations. So even when there is the outbreak of the dengue virus, so our government has recommended decoction for fighting against dengue, but we don't have anything of safety about Andrographis. So that if you have this kind of document, regulatory document, it will be good for the healthcare community. So we need kind of regulations to make sure that it doesn't cause any serious toxicity. So these are the preparations available as a supplement, mostly available supplement, not as a pharmaceutical compound. You can buy from online, you can buy from some pharmacy, but they are not a pharmaceutical compound. They're not approved by the Minister of Health or maybe our controller authority. These all available online, because they have been sold by supplement under the nutraceutical category. Can start from the and this the Calmac is the actually common name. So all the compound has been marketed in the immune booster. You can see it helps immune system and immune formula, immune fourth, so immune health, just for the general health. So in China, they have some tablet available, it's capsule available for immune as a dietary supplement. Okay, they are promoting as a dietary supplement. So for the cold, they have the antiviral. I mean, antiviral they are recommending as a capsule to immune defense. So these all as the supplements. They are not the pharmaceutical compound. So these are some of the patents has been filed by the different countries. I just want to quickly summarize. So you will find a lot of information here. So after making some changes in the structure, they found some improvement in the antiviral activity. In China has made 2 patents on the Andrographolide in 2014. One for the antiviral, it's a general antiviral activity, Andrographolide derivative. After making some modification Andrographolide, and this is the patent number and the year. There is another compound. This is a semi-synthetic compound, 14-deoxy. The other Andrographolide is for hepatitis B infections by the company. This is the patent number. And from the University of Singapore, they also made another patent, Andrographolide, for the inflammation and airway disorders. This is the US patent. These 2 are the China patent. And you will find all the information about what are the attempts has been made for the different activity. It is not just only antiviral. You can also see here different pharmacology activity. They tried different changes in the structure, and the pattern has been made. You will find all the information about these 2 articles. This is some of the very important pattern related to the antiviral activity. I'll show you. But now people are talking about the Andrographis and immunity. It was recently published in the magazine. So people are now started to think back. So why did you work on the Andrographis? It's a big opportunity for immunity to fight against the immunity, but it's still in the form of nutraceutical. There is no attempt has been made to develop the drug from the pharmaceutical aspect. We should focus more on the pharmaceutical aspect rather than the nutraceutical aspect. Just want to conclude. So the demand of the Andrographis every year. More research is going on. More people are attempting to work on the mechanism, try to isolate compound from the Andrographis. Okay, so the response is overwhelming response because the potential is really great, and it has a vast literature, traditional literature, okay, very traditional literature, old traditional literature, and this extensive pharmacological effect has been identified. It's up to you. You can interested. You can choose any particular activity. You can work on it. The safe herb is a highly important and medicinal plant for the general mankind. So it's very useful, particularly very useful for the immunological applications, like for example cancer, or can be viral disease. Based on the literature, I just wanted to conclude this point. Even though in terms of pharmaceutical aspect, we are still far away. So if you ask me how many products we have in the market as a pharmaceutical compound, so I have no answer for this one. Okay, we still far away. So I have to start working on this kind of precise herbs. Okay. So how our future direction could be helpful to develop a drug molecule to see the Andrographis in the pharmaceutical sector. We need a well-designed clinical study. I mentioned there was only one study has been done. Only one clinical study has been done in India. We need more clinical trials has to be done. It should be well designed to evaluate the effectiveness and also potential and also safety. These all things should be assessed in India, and all the pharmacology activity has been tested, must be validated. So not just only the antiviral, the rest of the other activity has to be validated through the appropriate clinical study. Unless we are not going to be successful, okay? When you do this kind of initiates clinical trials, which may helpful to reduce the antibiotic and antiviral dependency for the primary care. We can still use Andrographolide for in replacing the antiviral at the primary care level. Okay, that's one of the approaches we can get the benefit. So we need to explore on this clinical trial, and it could be really an alternative to antimicrobial resistance, or it could be new antiviral if you do properly in the clinical trials, we can come up with a new medication for to overcome the resistance of the antimicrobial, or it could lead to new antiviral compounds. And also, not just only about the pharmacology efficacy, the safety also should be considered, because the drug has some kind of drug interaction, so must be addressed. Okay, make sure that it does not cause serious issues. So these all the things to be done based on the clinical study. So, and we should start focusing on the commercial benefits. So we have some foundation, we have some data, some clinical data. We also got very good pharmacological information has been proven. And why did you go for some formulations? Okay, focus on commercial benefit. We are not focusing like China has a lot of formulation, a lot of capsule. They come up with a lot of formulation. They're looking for commercial benefits, but our Andrographis in India is not that much popular. So we need to have a novel strategy to develop a new formulation and development, and start working on the semi-synthetic analogs and new formulation, herbal formulation. Try to work on the new delivery system that the bioavailability problem. So we should work on some kind of delivery system to enhance its therapeutic efficacy. That's one of the major limiting factors. Not just doing only on the pharmacology activity. We should focus more on the commercial demands beside the pharmacology activity. So these are the two things we should address in the as a pharmaceutical scientist. So with this future direction, I wish to conclude my talk. Thank you so much for your attention. Thank you. Thank you so